to help with the disease and also the treatment. Okay, so that's the concept. Just a brief through about the personalized therapy. Okay, when I think same like other lecturers, I guess. Okay, most of lecturers that get involved with your teaching must be related to with the cancer research, if I'm not mistaken, right? So do I. Okay, like I'm attached affiliate to pathology department in School of Medical Sciences. So I'm a research lecturer working on this breast cancer and also colorectal cancer. Okay, but what I want to highlight here when we look just a brief idea about what happened in cancer cases nowadays is that you can see here there's a difference in terms of cases related to the less developed regions with the more developed regions. So you can see the new cases, okay, is increased in the less developed regions as compared to the more developed regions, okay, which we can see that the awareness about cancer is increased where people or local people, for example, they presented themselves at the hospital for the diagnosis. Okay, but then in terms of here, you can see the death here. The death rate in the less developed regions is much higher as compared to the more developed regions, which suggesting that more in terms of designing the treatment options for the people need to be done, okay, especially that can benefit to the people from the less developed regions. And also when we talk about the cases of cancer, it's not about cancer case. We expect that the cases is decreasing, okay, yearly, but perhaps what they predicted is that in 10 years time, okay, the cancer cases, it will be continually rising. So meaning that we need to do something in order to prevent the cases to get increased. Okay. All right. When we talk about designing the treatment option, designing the screening option for the cancer patients, it's all about understanding the cancer characteristic. Okay. So in here, what I put here to understand the cancer characteristic, you need to normally I'm not sure whether you're aware about these cancer hallmarks. Okay, where Hannah Wennerberg, a model. Okay, it has been proposed in 2000, year 2000, and it was plus minus, I guess, 10 years, 10 years ago, 20 years ago. Okay, so you can see how I think 20 years progression. Sorry, this one is about 20 years plus progression, where you can see how from the basic six cancer hallmarks, okay, based on the research that has been done throughout the years, they propose to have more than, about, I guess, about 13, 15 of the hallmarks of the cancer, meaning that the characteristic of the cancer has been well understood by time to time, uh, allowing that the researcher or the scientists to it helped the scientists to design the what kind of treatment that we can do, especially to benefit for the patients. Okay. So far so good. You can stop me if you have anything to ask. Okay. All right. So our topic today is about omics. Okay, omics. So basically, omics, it's about based on one of your colleagues just now. They said that about the collection of data, integrative data, coming from genomics, proteomics, so whatnot. But you can omics here is actually based on the integration of big data. Okay, because of just now, remember I mentioned about how we want to achieve that personalized medicine, how we want to build up a model that can actually give a comprehensive data about the patients. Okay, so we have to cater many angle. Okay, from the gene side, from the ep, epigenetic sites, transcriptomic sites, protein sites, as well as metabolome sites. Okay, so you integrate all the data, and then you can actually give a sort of like a big model, a good model, to understand the treatment option that is available for that particular subgroup of the patients. Okay. All right. So I'm working at pathology, okay, as a research lecturer here. So in anatomical pathology, okay, let's have a look on the rationale why we need to have this multi-omics data, basically. Okay. So in pathology, okay, what we did is that definitely we study on the pathological conditions, the disease parts. Okay, so basically we depends on this microscopic observation. Okay, so basically what we did is that in here, with the H&E staining. Okay, this is actually H&E slides. Okay, H&E slides. So you can see from here that based on the H&E technique, you can observe the histology of the tissue. Okay, this is the tissue, the tissue sections, when you can study the morphology of the tissue. Okay, so based on the morphology, then you can subgroup the type of the tissue. Okay, you can give a grade on that tissue. So for example, in here, for example, for the breast cancer, okay, they got the histological grade from grade 1, grade 2, and grade 3, okay, based on the microscopic observation. Okay, and then you know that with this H&E, it's just based on two colors, the pink, the blue, the purple. So basically you need to be an expert to understand about the changes behavior, okay, the behavior changes at the surface level, okay, at the cell level. But then if you require more understanding, okay, you may apply some other technique, the so-called spatial technique. Okay, when you can locate some more what do we call that, staining on the other area of rather than nuclear, cytoplasmic, and membranous. Okay, so for example, this spatial staining, it stain this so-called adipose tissue. Okay, so with this when you apply a special chemical, okay, that chemical will provide another color that will specify the location of your interest interest location, like adipose area here. Okay, but in pathology also, we apply another technique where we call as a immunohistochemistry. Okay, immunohistochemistry, where I'm sure that you guys are aware about this. Okay, this technique, immunohistochemistry, very common technique to study on the protein expression. Okay, you have your interest antibody. Okay, you stain at the tissue level, at the tissue surface, and then this brown color indicate your interest protein expression. Okay, with this brown, the DAB expression. So basically with immunohistochemistry, you can see the protein expression, okay, whether it is highly expressed or it is low expressed at that particular tissue. Okay, and to somehow, when you want to have more than one interested marker, okay, interested protein, for example, you may need to apply another technique where we call as immunofluorescence. Okay, this is immunofluorescence. Okay, you have a special fluorescent dye to remark the expression of different protein. For example, they got like two type of protein here, the lamin B and also the choline. Okay, so you stain them. So basically with the immunofluorescence microscope, you can see the location, okay, where the protein get located and how much the protein get expressed in that particular area, which is good actually. And then you can actually merge them in here to see whether the merge is actually can indicate some kind of co-interaction, co-expression between marker A, lamin B, as well as marker B, choline. Okay, so this is some kind of a basic technique that being done in pathology. Okay, so for example here, when we talk about targeted therapy, therapeutic approach, right, in our topic today, so it's a well-known target. So for the breast cancer, okay, with the immunohistochemistry, you look at the level expression of ER, estrogen receptor, PR, progesterone receptor, as well as HER2. Okay, based on these three important marker, you can actually the clinician, okay, the oncologist, they use this as a very important information for the treatment option. So for example, in here, based on the ER, PR, HER2 expression, they characterize the type of the breast cancer here. So from luminal A, luminal B, HER2, as well as triple negative. So you can see that from here, from luminal A, it can be considered as a better prognosis because it got ER positive, HER2 positive, PR high, okay, and then up to the triple negative where they lost all HER2, ER, and also PR, which this condition can be considered worse, okay, to decide for the treatment itself. Okay, so when we talk about treatment, okay, here you got PR, progesterone receptor. Okay, progesterone receptor, they design with the estrogen receptor, they design treatment, the so-called fulvestrant and tamoxifen, which is block the expression of ER and PR. Okay, and also for HER2, they got the monoclonal antibody of trastuzumab, or the so-called Herceptin, which inhibit the expression of the HER2, the activation of HER2. Okay, so this is actually the treatment that I'm showing here is actually a well-known treatment for breast cancer, and it is considered as a targeted therapy. Why targeted therapy? It's because of they got the target here of the ER, HER2, and PR. Okay, so that's what happened in previous years about designing the targeted therapy for breast cancer. But the problem now is not all patients will benefit on this particular treatment. Why? Because of a group of patients that develop tumor resistancy, which is actually decreased the treatment efficiency. So why is that happen? Okay, why is that happen? So because of we need to understand on the concept of connection between molecule to molecule. Okay. So when we talk about new era of anti-cancer therapeutics, okay, sorry because of I'm using cancer as an example here, okay. So we need to identify the drug targets for cancer treatment. Okay, so if previously they rely on the surgery, when we talk about cancer therapy, they rely on surgery, radiotherapy, chemotherapy, and also immunotherapy. But of course, with this therapy, it provide low response rate and also multi-drug resistant, as what I mentioned just now, and recurrence or metastasis and also target mutation. So these are the challenges happen so far in designing the anti-cancer therapeutics. So what we need to have now is not only to design a targeted therapy, but we also need to design the personalized precision medicine for cancer individual, okay, which ideally increase the sensitivity of the drugs, improvise the drug resistance. So that's the concept behind that about therapeutic approach. Okay. So far so good. So far so good, or you guys?
Sorry, Doctor. Like the slides with us, it's stuck in the first slide that you explained for us.
Huh?
The slide on our like screen, it's just the one like personalized cancer therapy.
Personalized of cancer. Oh my God, you guys should
Yeah,
tell me.
we chatted, but I think you haven't seen it.
You guys know what? You know what? I already at the slide number 6. That's
No, we
why I
are
put
still in
you.
3.
But you still can hear me, right?
Yeah. Thank you.
Next time, if you notice that your lecturer is talking not related to the slide, let me know, okay? Meaning that I already at the slide number 6. So you guys still cannot see slides number 2 here.
Uh, Only the third one, the one personalized cancer therapy.
Personalized cancer therapy. My God. Here, still talking about personalized cancer therapy. Okay, what about here? What about this one? Is still moving? Not moving. What about now? Is it moving?
Uh,
No.
Doctor, no, but you changed it, and then came back to the same.
Came back to the same. And then, okay, when I change here, I change the slide. No, cannot see that. Still freeze. Oh my
There is
God.
no slides, doctor.
There's no slide moving in. What should I do then? Oh, okay. Hang on, yeah. We try, yeah. Screen window. Okay. Okay. Also, is this still the same? You guys still?
No, doctor, it's working.
It's
It's
working
different
now.
cancer hallmarks.
Yes. So you haven't seen this slide before.
No, doctor, one of.
So it seems like I was talking alone. Okay. Okay. I changed these slides to the next one. I don't know. Is it moving or not? Okay. What about now? Is it moving?
Yes, doctor.
Okay.
Change.
All right. Okay. Where should I start then? Okay. Just now we are talking about personalized cancer, and then we you remember that I'm talking about the rationale of having these cancer cases. Okay. Or you guys need me to repeat that again?
Maybe you should do
Repeat.
and it would be easier.
I feel bad. Okay, all right. So just now remember that we talk about the personalized cancer. Okay, and then personalized cancer is about to subgroup the patients to allow the clinician for the treatment options. Okay, for the oncologist. Okay, now the rationale behind that, why we need to do this, because of you look at this cancer incidence. Okay, they got two types here: the incidence at the less developed regions, more developed regions. Okay, so you can see that nowadays the new cases in the less developed region is increased. Why? It indicates that the cancer awareness is good, is better nowadays among the populations. Okay, so they presented themselves at the hospital for the cancer diagnosis. But then when we talk about the death incidence, okay, here in the less developed regions, okay, it's pretty much higher as compared to the more developed regions. Okay, so this indicates that we need to think about how to design a therapeutic approach that can actually benefits to the local population on that particular regions. Okay, so definitely when we talk about the cancer cases, we expect that the incidence is decreasing by year, but perhaps what they expect predicted in 10 years time, the cases is going to continually rising. So meaning that we need to do something in preventing the cancer incidence. Okay, all right, so Can you see this slide? Guys, can you see this slide?
Yes, doctor.
Okay, so this slide is about cancer hallmarks. Okay, so you can see that, sorry, this is actually 20 years, 20 years progression. Okay, 20-year progression. So Hanahan Wennerberg, okay, has proposed this model. Initially, they got the 6 characteristic of cancer. Okay, so yeah, because just now we're talking about treatment option, designing a treatment option for the patient. So definitely, to design that, we need to go to the basic things where we need to understand the cancer characteristics. So, for example, with the cancer hallmarks previously, in 2000, they got the 6 characteristic about cancer. Okay, and throughout the years, okay, after 20 years time, okay, you can see that the characteristic has emerged, okay, from 6 and become, I guess, more than 10 here, more than 10, which allow the researcher, the scientist, the clinician, and to to understand, okay, when want to design the treatment, okay, to that particular patients or to the group of the patient. So these are the characteristics that you need to be particular with. Okay, that's about cancer hallmarks. Okay, come to think about the omics. Omics. Okay, omics just now, all of you answered that very good, okay, about the understanding on the omics. So omics is about you are generating big data. About what? About that patients, about the cancer characteristic, about the patients. Okay, you take it from any angle. To get the data, you take it from any angle, from the gene side, from the RNA side, transcription, from the epigenetic sites, and then you got from the proteomic sites, protein sites, as well as the metabolite sites. Okay, so this integration of multi-omics data kind of giving us the very comprehensive understanding, okay, about what is actually happened in terms of the behavior of that particular cells. Okay, so I'm working at pathology. Okay, see, this is what happened. Okay, I'm sharing you some side about what is happening in anatomical pathology. So in pathology, everybody knows that we study on the disease progression. Okay, so we are doing this based on this microscope. Okay, you can see the slides, right? So far, so good.
Yes, doctor. There's slide.
So no, what's no here? No, nothing. Yeah. Sorry, I just have a I look at the side of my slide at the moment. Okay, so I cannot see your chat at the same time. Okay. All right, carry on with that. Okay, so just now I mentioned about anatomical pathology where we observe using microscope, right? And then okay, so for example, this is an example of the tissue section of the breast cancer. Okay, so based on the H&E staining. So this is H&E staining. Uh, you Google it, is hematoxylin and eosin slide. Okay, H&E. So they stain in the nucleus, cytoplasmic, and membrane. So basically, with H&E, you can distinguish the location of the nucleus, cytoplasmic, and membranous. Membranous is yes, cytoplasmic and also the nucleus. And then you have an expert, okay, pathologist. They will actually study this behavior of the cells, okay, behavior of the disease based on the changes at the tissue level. Okay, so basically, for example, breast cancer, okay, they can subgroup them into grade 1, grade 2, and grade 3 based on the histological grade. Okay, and perhaps when you study H&E, it's just based when you study the histology, it's just based on the changes at the surface level. Okay, but what you, of course, you may need to have more information, right? Okay, so definitely you can apply some additional staining, for example, special staining, where you integrate add on a certain chemical substance, and then they will locate your interest region, such as adipose tissue, so on. Okay, and then there's another technique, we call it as the immunohistochemistry, ISC. ISC, when you study the protein expression. I'm sure that you have learned this in before. Okay, immunohistochemistry, ISC, you study the protein expression when you have the antibody antigen reaction. You have a special interest. You have your interest antibody on the, and then you look at the level expression of your interest antibody or protein expression based on the brownish color here. Okay, so ideally with the ISC, you can get the idea somehow the protein is highly expressed or low expressed. Okay, so this is, but somehow you may need to have more than one interest antibody where you will apply this immunofluorescence. Okay, so immunofluorescence where you can have two targets here. Okay, for example here, laminin 2, which is your target number 1, and then choline antibody target 2. So basically with immunofluorescence, not only you can locate the expression, but you can also see how much the level of the protein get expressed. And perhaps you can also merge them to see whether your interest target is actually co-expressed together or not. Okay, by looking at this expression. So that's what basically some idea what happened in the diagnostic technique for the in the pathology, anatomical pathology. All right, so okay, do you get this slide? Yes or not? Before I start, can you see the slide?
Yes.
The new one.
Yes, doctor.
Okay, sorry. Okay, I have to keep asking you. So this is a well-known targeted therapy. So if you remember what I'm saying here, for the breast cancer, normally these three important markers are very commonly used so far: ER, PR, HER2. Okay, so they based on the immunohistochemistry, we look at the protein expression of the ER, PR, HER2, and with the level of ER, PR, HER2, it will help in terms of determining what is the best targeted therapy to be given to the breast cancer patients. So, for example, you can see here the subgroup of the breast cancer based on the different expression of ER, PR, HER2. So, for example, ER positive, HER2 positive, PR positive, they consider as luminal A. Okay, and in fact with the PR positive, even though it is high or low, it will distinguish between the breast cancer type of luminal A and breast cancer type of luminal B. So basically with the different type of breast cancer, you can see whether it belongs to the better prognosis or worse prognosis. For example, triple negative here, which is it got negative expression of ER, negative expression of HER2, and also PR, which considered as worse because of we need to think about some other treatment option for that particular patient because of it no longer rely on the ER, HER2, and also PR. Okay, so we talk about treatment here. You can see that tamoxifen, fulvestrant are the common treatment given to block the estrogen receptor and also the progesterone. Whereas trastuzumab, or we call it as the Herceptin, help to inhibit HER2 expression. So ideally, this is considered as a targeted therapy because they have a special marker to target with. Okay, they design the treatment based on that particular marker. But the problem is that with this targeted therapy, it does not. It cannot give a good 100% efficiency, 100% it cannot be 100% efficient to the patient. Why? Because of some group of patients that develop a tumor resistance, okay, which ideally can decrease the treatment efficiency. Okay, all right. So moving on to the next slide. Okay, so when we talk about this in the new era of anti-cancer therapeutics, okay, Um, I've been explaining before. Okay, what so far when we talk about cancer therapy, so it relies the common therapy agents given is surgery, radiotherapy, chemotherapy, and also immunotherapy. Okay, but perhaps this therapy they bring into some challenges. For example, low response rate, multi-drug resistant, and also recurrence, and may need to have more target mutation. Okay, so in designing the anti-cancer therapeutics, we need to identify what is the target here. Okay, and then we need to design towards on personalized or the so-called precision medicine. Okay, and also ideally we need to increase the sensitivity of the treatment as well as to improvise the drug resistance. So that's some rough idea about what is happening in terms of anti-cancer therapeutics. Okay. Mm, but yeah. Okay. When we talk about resistance, right? Just now, I've told you, the ER they've got like target here, the ER, PR, and also HER2. Okay, but perhaps they still some tumor they develop resistant towards on that treatment because of the rationale is you can see here HER2 for example. When we study HER2 for example, HER2 has this they got the marker itself has a different type of markers. The siblings, okay, siblings. So for example, HER2, it has HER1 and also HER3. Okay, they belong into one family members. Okay, so ideally, HER2 is a marker that presents at the surface level. That's why um, when you perform immunohisto, you can see the protein expression of HER2. Okay, with the immunohistochemistry just now. Okay, but when with the activation of HER2, once it get expressed, so normally it will activate the downstream channel of the PI3K, mTOR, and also RAS, ERK pathway, where this actually helps to turn on the signal for the proliferation, survival, invasion, and also angiogenesis of the cancer characteristic itself. So ideally, you can see that with the HER2 activation, it helps to progress the cancer. It helps to develop the tumor progression of the breast cancer, for example. Okay, so when you target on HER2, ideally, you cannot say that it can be 100% um, give a good result. Why? Because of you can see the nature of HER2 itself. It exists in the different it need to it need to co-partner with some other receptor. Okay, so for example HER2 and HER3, HER2 and HER1. So ideally when you block HER2, so HER1 and HER3 is still there, which can still activate this downstream marker, this downstream signal that helps to support the tumor progression. So you can see this slide, okay, this picture, actually, they design a treatment, okay, that inhibit each of the signaling. For example, PI3K, they got this BKM120. The HER1, they have this lapatinib, naratinib, that targets on each of the receptor. Okay, ideally because of they need to increase the sensitivity in killing the tumor cells. So that's about um, how they want to tackle the issue of the tumor resistance. Okay, so when we talk about that, definitely not only we need to rely on the surface level, we need to go deeper on the cell itself. You need to go deeper on the cells to look at some other molecular components inside the cell. Okay, so basically, when we talk about components in the cells, okay, molecular components. So ideally, you guys know about this. We got DNA, you got the RNA, and also protein. So basically, DNA, RNA, protein, they are located at a different localization of the cells, right? For example, DNA, it is located in the nucleus, RNA outside from the nucleus, and the protein is the one that located near to the surface membranous, okay, at the outer layer of the cells. Why? Because of the protein is the one that will control the behavior of the cells. Okay. So what is the rationale of study these molecular components? Okay, again, tell me, how many of you know about central dogma or heard about this? You have no idea what is central dogma? Have not heard about this?
Yes, doctor, we have learned this one from the first class that we had in our master.
Okay,
Yeah,
that, that's cool.
it's about our biological information, like coming from the nucleus, from DNA, and then to RNA, and then from to mRNA, and then producing that proteins. So
Right.
right now all the focus and also like our course is based on this idea, like molecular medicine. So it's go back to all the molecular mechanism and molecular
Exactly.
aspect of cells that happening with the disease and cancer and any kind of the disorder that human facing right now.
Yeah. Okay. Thank you, Sana. All right. So, okay, Jai say heard. Okay. Mm, okay. So basically, basic basic explanation why we need to understand about molecular components for me, okay, in my opinion, the how you want to transit the components, the molecular components from one location to another location. So for example here, because of in previous slides I've shown you that different molecular components, they are located at different localization of the cells, right? So basically you can see the DNA, for example, here it consists of a very long chain DNA. Okay, so ideally, how it needs to be transported out from the nucleus. Okay, so there is a mechanism where we need to simplify the sequence of the DNA, okay, and then become the RNA here. Okay, to undergo the process of the transcription. Okay, so the transcription is they copy the whatever sequence from the DNA to be at the RNA, and then RNA become a single strand. Okay, so you can see from here the RNA itself, it consists of the region that have translated coding, the coding region versus the non-coding region. So now we're talking about coding region because of in order to understand the cellular behavior, we need to have that coding region. Okay, so what we don't need is the non-coding region. So of course, can you see that from a very long sequence of the RNA, it need to be chopped off into a very small chunk, in the big chunk to become a smaller chunk, smaller part, and then they remove out the introns. Okay, undergo the process of splicing. So what happened here is that based on the process, so they produce the so-called messenger RNA. So can you see that the sequence now, right now, from the big chunk has become a smaller fragments, which can be easily to be transported out from the nucleus to the cytoplasmic. So at the cytoplasmic, so there you go, the another process of translation happened. Okay, from the mRNA to become the amino acid chain. Okay, so here they undergo the process of translation. In this process, translation, so you can see that how they need to have the transfer RNA, okay, and also the ribosomal RNA. So it's kind of how to, this is more like a process, copy editing. Copy edit, not copy editing. It's like this one, the ribosomal RNA, they will undergo the sequence of the messenger RNA, and then they try to read out, and then they translate, read and translate. So the transfer RNA is the one like a print, thumbprint, to read the codon and then translate to become the amino acid chain. Okay, so you can see that at the end, the protein is synthesized, and at the protein level, So we can basically understand whether whatever changes happen in at the DNA, it can be translated or it can be functional at the cell level or not. So that's the idea about understanding the molecular components. Okay, so this is genome transcriptome. Okay, so basically just now, as I mentioned that the DNA is a very big chunk, okay? But what we really need to have is a very small part here, the coding RNA is the one that will be the mRNA translated into the protein, okay? But do we need to ignore this transcriptomic that consists of the non-coding RNAs? Okay, we cannot do that because of this thing is very important for the modification of the mRNA, which I will explain to you a bit later. Okay, now we talk about omics. Okay, we talk about omics. Okay, it refers to the genomics. It refers to the study of the person's genome. Okay, where the epigenomics, okay, is the study of the complete set of epigenetic modification of the genetic material of a cell. Transcriptomic is the study of the RNA transcript produced by a genome. And then proteomics is the study of the proteomes, a set of proteins in an organism. Okay. Metabolomics, study of the metabolites, okay, which is the small molecules within a cell that produce during metabolism. Okay, so that's a basically definition, but I believe that you guys have known about the definition of each of the omics here before. All right. Yeah. When we talk about the integration of the big data, there you go. Nowadays, they call it as a single cell omics. Okay, single cell omics where they use this approach. Okay, all of the design. Okay, to understand about the behavior of the single cells. Okay. Oops, sorry. Give me a minute. Okay. Okay, so with the omics thing, it's kind of a bridge that you need to understand between underlying molecular changes and also the cell behavior, which facilitates a deeper understanding of the disease. In my case right now, I focus more on cancer development. Okay. Uh, the importance. Okay, the application wise, definitely we're talking about, I'm sure that before this you have learned about the biomarker discovery and then the screening diagnosis, for example. But my case right now, we need to look at from the aspect of the therapeutic targets. Okay. Okay. This is a prospective clinical studies guided by omics data. All right. So basically, like what I said, this is a patient cohort. Okay. You generate the data from the cohort. Okay. And then you classify them. Okay. Into based on their profiling just now. So they are doing DNA sequencing, for example, gene expression study, copy number profiling. So all of the data that you generated from the cohort will actually subclassify them, okay? Or you can use that as a personalized individual profile, okay? And then you bring to the molecular tumor board. Okay, you use whatever data that you have here from as a biomarkers to assign treatments. You use the gene drug relationship. Okay, if there is no biomarkers that are available. Okay, from these, you will select what is the best therapy. Okay, based on the relationship between the drugs, the genes, and also the tumor risk. Okay, they got three things here happening: drugs, genes, and also the tumors. And then you will provide the treatment of the matched patients. So this is the off-label drugs. What is the off-label drugs? Off-label drug is drugs that has been a targeted drugs, I would say, but a targeted drug that has that does not have the non-FDA approval. Okay, the off-label drugs, they provide based on the treatment decision from the profiling here. And then remember, they need to see the effect. Okay, so this is based on the survival data. Okay, so based on the survival data, then you can see ideally whether treatment is functioning or not to that particular cohort. Okay, so that's this is a basic idea what happened when you apply the omics data in the clinical study for the treatment options. Okay, all right. So just now you can see what happened in that clinical prospect, but then when we talk about in the lab wise, okay, how we are going to generate the omics data. Okay, so just now you understand about multiple approach here: genomics, epigenomics, transcriptomics, proteomics, and metabolomics. And definitely different approach will have a different components. They will utilizing a different components such as genomics. They use DNA, epigenomic modification of DNA, DNA methylation whatsoever. Transcriptomic, they use RNA or they transcribe into cDNA. Protein for proteomics, metabolites for the metabolomics. Okay, so that's about the components being utilized for the different omic approach. Okay, so basically when we talk about technology here, so to understand, okay, you utilize the DNA. From that DNA components, you need to provide these results. Whether the mutation happened, is there any genetic variants? Is there what happened to the gene presence or absence? What happened to the genome structure or not? So to understand all of this, you need to use this technology, sequencing or exome sequencing. Okay, and then modification of DNA, DNA methylation, for example, DNA modification. So you use PCR, qPCR, and somehow sequencing also can be applied. Mass spectral also can be applied in this method. For the RNA and cDNA, okay, in transcriptomic, okay, in order to understand whether whatever mutation happens at the DNA level, it expressed or not, you need to have this RNA. So you study the gene expression, you look at the whether it is highly expressed, positively expressed, or low expressed, negatively expressed. So all of this you may need to apply the technology of PCR, whether it is qPCR, RT-PCR, so whatnot. Okay, and of course you can also study the sequence of the RNA by applying the RNA sequencing. Okay, so same goes with the protein. Protein, remember that protein is in term of the structure. So protein must be very complex, okay, as compared to DNA, RNA. So with the protein, you still can study the sequence. Not only you study the protein sequence, but you can also, at this level, you can start looking at the interaction between target protein 1 with the target protein 2. Okay, and this can be done by applying some technology of mass spectral, okay, methods of Western blot, and also ELISA. So that's what happened at the protein level. Okay, same goes with these metabolites. Metabolites also you apply some of the advanced technologies such as mass spectral, NMR, spectroscopy, as well as HPLC. Okay, so that's about the technology you use to to generate the omics data. Okay, so this technology we consider as a high throughput technology. Okay, so for example, we go to the very common one, next generation sequencing. May I know how many of you have performed experiments on, have get involved with this technology, NGS? Hmm? No, 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 no. No, no. Okay. Any idea what is NGS? No. Also no idea. Okay. So NGS we call it. Yes, we have lectures on the previous semester. Oh, good, yeah. Okay. Mana tadi? Okay. All right. So this, yeah. So I'm sure that if you have lectures previously, so you got some ideas what is actually the principle behind the next generation sequencing. So ideally, with the sequence, it is a next generation sequencing. Um, how can I say this? It's the next generation sequencing. I would consider as an advanced technology after the direct sequencing, the direct Sanger sequencing. Okay, so we will see the difference later at the end of my lecture later. Okay, so basically with the NGS, they will divide into three things. Okay, genome sequencing, where you scrutinize, you sequence the entire genome, okay, of the species. All right. So you cover all genes, you cover non-coding DNA, okay, but in terms of accuracy, because of you cover all, it it can be considered as a low. Okay, and definitely when you need to entirely sequence the genome, it may require longest turnaround time. And of course, the cost must be very expensive. But in terms of the depth of the understanding, okay, of the sequence, it's much, much higher. Okay. Um, that's why, okay, from genome, they revise the technology, design the so-called exome sequencing. Why exome? Remember the concept of the transcription just now? Okay, you exclude the non-coding region. Okay, what you want to have is a coding region. Yeah, so you just selectively get the region of the exon 1, exon a certain exon, and then your entire gene you sequence the exome. Okay, the accuracy is better as compared to genome sequencing. Still, provide long turnaround time, um, cost effective. Okay, and also the depth is much better as compared to the genome sequencing. Okay, but targeted gene panel has become the chosen one right now because of in the clinical studies, for example. Medical practitioners, okay, oncologists, pathologists, they already have a specific gene panel that they want to know because of that panel has been considered pathogenic to one particular disease. For example, cancer. Just now I've shown you breast cancer, for example. So we got ER, PR, and HER2. Okay, so that three gene is already considered a pathogenic gene panel for the breast cancer. Of course, there are some other gene that included into that gene panel. Okay, so that with the targeted gene panel, the efficiency is much better. The efficiency of the sequencing is much better. Okay, even though the coverage is not very wide, very big as compared to genome and exome, but the accuracy is much higher. Okay, because of we have a specific gene panel, and it will provide the rapid turnaround time and also most cost effective. Okay, in term of, this is about the difference between genome, exome, and also targeted gene panel. Okay, so this is I put the picture about the flow work uh, of these, what do we call that, the NGS. Okay, the sequencing. Okay, this is a sequencer. Okay, basically you start with the library preparation, and then with the library preparation is when you index them. Okay, that library provision have some kind of adapter. Okay, and then you sequence, and from the sequence it will link to the computational tool for a data analysis, and then you will interpret and give a report for the final results. Okay, so that's about next NGS. Okay, and then instead of NGS, okay, NGS, they are very popular for these so-called analysis among solid tumor. Okay, and also these, what do we call that, the blood tumor. Okay, but somehow there is some other samples that may require another technology to apply, which is the so-called cancer liquid biopsy. So in this case, okay, droplet digital PCR is better, okay, to have a greater sensitivity to detect targeted mutation and also cost effective, right? So for the liquid biopsies. Okay. Another technique, remember that I've been in the previous slides, there is a table of the about the technology. So another technology is on the qPCR, okay, where you utilizing the RNA component. Okay, with the NGS, you utilizing DNA sequence. With this qPCR, you look at the level expression of the RNA. Okay, and then you use qPCR somehow. qPCR is actually the advanced technology coming from the basic conventional PCR. Okay, so for example here, so you basically with the PCR, you get the results at the end of the process where you load your samples, okay, your PCR samples into the gel. So basically with the gel, I don't know whether you can see this or not. Okay, with the gel here, so you can see the bands. You assess the results of your PCR based on the bands. Okay, based on the bands, whether it provide a good intensity, low intensity. So basically with the PCR, you can only observe whether it presents or not qualitatively. Okay, but nowadays they change, they revise the technology, they provide the technology that where you can access the amplification by the time, throughout the time of the amplification of the PCR process of the cycle. Okay, so you basically see which cycle that actually the PCR start to get active. Okay, provide the until it achieve into the plateau cycle. So based on the results here, okay, based on the technology, they use a fluorescent. So the fluorescence actually will provide the value, the number, the reads that you can get. You use a certain formula to provide the value of the targeted expression of your RNA, targeted RNA expression. So this is an example of the relative expression. So you use a control, okay, you use your control and then you provide the relative expression, and basically you can say whether your target gene is decreased, low expressed, or highly expressed. Okay, so that's a concept from the conventional PCR to the qPCR. Okay, so some other methods being applied so far to study the RNA expression is microarray. Okay, with the microarray, you can actually look at the level expression using abundance of targeted RNA. Okay, so you use a chip. Okay, you hybridize your cDNA probes to oligo sequences on microarray chip, and then you scan, and then based on that, it will provide the level expression, your level expression based on this, the color dyes. Okay, so for example, the results like turn out to be like this. Okay, where with the microarray you have these gene expression patterns. Okay, gene expression patterns. Remember that previously we the classification of the breast cancer is only based on HER2, and PR. But nowadays with the microarray, you have more target. So when you have more targets, so you can precisely subclassify your breast cancer into into a different expression, into the different group: basal-like, HER2, normal breast-like, luminal C, luminal B, and also luminal A. Okay, so this is what we call gene profiling based on the microarray. Okay, so far so good, everyone. Any question? No question. Okay. All right. So let's move on into the, I guess this one we move on into the application, right? Ah, okay. All right. Come back to this concept. Okay. Just now, okay, you based on the slides, these slides, okay, the NGS, droplet digital PCR, qPCR, microarray. So ideally, I think you already know that, okay, just to get some idea what is actually the advanced technology available so far, okay. But then when you talk about omics, remember, you need to generate more data from a different approach at one time. So ideally, your technology need to be advanced, okay. So need to be advanced in terms of you need to do it very quick, faster, generate, and also generate much comprehensive data. Okay, so when we talk about the principles of multi-omic measurement, okay, from a single cell, okay, you need to design a study to target not only one component such as DNA or RNA, but you need to do it together. Okay, you need to have a DNA as well as RNA as well as protein all together, and then run it into using one technology platform, and you will generate much intense data from that analysis. Okay, so in principle one we call it as physical separation. Okay, physical separation meaning that in physical separation you have these from one single cell. Okay, you lyse them. Okay, and then you will release the DNA and RNA. Okay, from there you will integrate the bits, the oligo-DT coated bits. You coat the bits, okay, and then you apply the so-called magnetic pull down, okay, to separate between the DNA and also RNA. So once you got that RNA and DNA, so ideally with the RNA, okay, you will straight away analyze for the RNA sequence, DNA for the DNA sequence library. Proceed with the sequence library. So can you see that you don't have to do it repetitively. You don't have to repeat the experiment, okay, using the same single cell. But with one single cell at one time, you already have applied one approach where you can utilize the RNA and DNA at the same time. Okay, so with this, hang on, yeah. Oh, sorry. So what's the uh, advantage of having this method is that it can be used to complete the cells and nucleic from the both fresh and frozen tissue, and it can be processed further with DNA or RNA. But the problem with genetic is that RNA and DNA molecule might be lost during the physical separation. So in here, okay, you integrate the bits and then you need to do the physical separation. Okay, so ideally, you may lose the components of DNA or RNA throughout the process. Okay, so same goes with the B. Okay, the principles number one under the physical separation is when you use you lyse the cell. Okay, but then not of the nuclear membrane. Sorry, this is membrane. Lysis of the cell membrane, but not of the nuclear membrane. and then you do the physical separation of the nucleus from the cytoplasmic using the precipitation. And then when you do nucleus precipitation, ideally you can separate the supernatant, okay, aliquots. I guess this is a cell palette that represent the DNA sequencing. The supernatant aliquot represent the RNA molecules where you can use for the RNA sequence library. Okay, so this is two methods to be considered under the physical separation about how you want to extract the molecule components, okay, to be done for the multiomics measurements. Okay, so can you guys see this, the GNTC SC charm sig SCN?
Yes, Dr. Roll. Yes.
Guys, do you hear me?
Yes, Doctor.
Yes, okay. So can you see, can you, okay, do you have any idea what is GNTC?
No, Doctor. Like it's just like it's
You
new.
have no idea.
Yeah,
Yeah,
no, I don't know.
XIDR6. Okay, so this is actually the abbreviation of the technology. So, uh, okay, can we do an assignment? Okay, very simple assignment. Okay, so whatever abbreviation down here, can you guys wrote to me the full abbreviation and also the function of this to me? Just put
So,
it in a table. Something like
sorry, Doctor. Like you want us to know the function of this gene?
not the function of the genes, to know the abbreviation. You know, this is the
Abbreviation.
abbreviation. Abbreviation. Yes, abbreviation. Okay, abbreviation. Okay, give give a full name of each abbreviations noted in slide 29 up to 33. Okay, boleh? Can we do it? This one. Give a full name of the function and state the function of that technology. I'm sure you have learned this right before. Okay. Boleh tak? Yeah, boleh tak pula. Can we do it? Okay, can ah? Okay, let's move on to the next slide. Okay, and okay, okay, just now is a principle physical separation. Okay, this principle, they are using the so-called joint preamp with the poly A RNA. Okay, why? Once they release, okay, to distinguish between the RNA and the DNA molecules, they add on this so-called poly A RNA, because the poly RNA will add on at the tails part of the RNA, which help in terms of modification of the RNA. So at the end, they will split the pre-amplification reaction into two. Okay, one's for the RNA-seq library, and then another one is for the DNA sequence library. And then the third principle is this is more like a NGS applied the application in the NGS technology. So where they use the barcoding principle. Okay, this barcoding will be joined in the library preparation. Okay, this barcoding, they have a separate barcoding for the RNA, barcoding for the DNA, and then this barcoding is actually will link with the computational tools. Okay, so once you run with the NGS, for example, so this coding will actually link to the indexes, and then we'll call out whatever call out variants they will link with the computational platform, okay, to be analyzed, to be further analyzed. So that's about the principles with the sequence split. Okay, and then the next one is they because of you need to have a target, not only DNA, RNA. You also need to combine with the protein. Okay, so you need to separate the molecular analytes, which is molecular components here, using a separating the combinatorial indexing. Okay, how they did that? Okay, with the cell, they subclassified the cell. Okay, decoding the cells, they barcode the cells. Okay, into a different chamber. They differentiate into the differentiate chamber, and then they barcode the different cells is there, and then they pull in together, and then they add another specific barcoding, okay, to again to have another cell specific barcoding, okay, on that molecular analytes, okay, and then from there they will start doing a further analysis. Okay, so this single cell or single nucleus multiomic is possible when the ideas of principles 2 and or 3 are combined. So meaning that once after they did this, they will apply another principle of principle 2 or combine with the principle 3 in order to generate more data. It sounded very complex, but it is what it is because of at the end, we need to have the omics and in the sense of omics data. Okay, so can you see here? In here, they changed the term of SAI, L3 DNA RNA co-assay, SAI CAR sequencing, and snare sequencing. Okay, so I need you guys to do some research on it. Okay, because of it's very important for you guys to understand the concept related to the omics. Okay, now we move on into the application wise. Okay, I'm sure that you guys um, got too much information regarding to the omics data. Okay, so with the KRAS mutation, okay, this is the application wise. Okay, so again, I take it a very well known marker. Okay, the so-called KRAS. Okay, the gene KRAS. Okay, the mutation on the KRAS has been widely studied on several types of the solid tumors: pancreas, colon, lung, uterus, thyroid, testes, stomach, bladder, as well as breast and liver. But of course, the top three is pancreas, colon, and also lung for the KRAS mutation. Okay, so ideally, okay, conventionally. Okay, remember I mentioned about Sanger sequencing before. Okay, so